Hi. So I'm going to just guess here that everyone has some kind of experience with diarrhea. Diarrhea can range from a mild inconvenience to cause of death within hours if left untreated due to dehydration. I study a particularly severe form of diarrheal disease called cholera, caused by the bacteria Vibrio cholera. When you this bacteria is prevalent, and there are millions of cases around the world annually, and it is most impactful on children under 5 due to current vaccines being very ineffective in this age group. When you're exposed to this bacteria through infection or vaccination, you produce anti-cholera antibodies. These antibodies protect you from full-blown disease. I'm interested in how this actually occurs. We and other labs have shown that antibodies are thought to protect you by binding to bacteria, which are hypermodal. They swim very, very fast. The antibodies bind to the sugars that coat the surface and render them completely immobile. They are dead in the water within a minute of antibody binding. These bacteria swim 10 body lengths per second. They're very fast, and they're immediately not swimming anymore, which is very impressive. And this is thought to be the main thing that drives protection in disease. While this sounds like we figured it all out, we know what's happening, largely all of this is a black box. We don't actually know how any of this is actually happening. So my project is trying to figure out how these bacteria go from modal to non-modal in the presence of antibody. In order to address this question, I created a library of bacteria that had thousands of different random mutations and tested each individual strain and how it responded to antibody binding. We're hoping we find at least one bacteria that still bound antibody but no longer undergoing motility arrest, because that would mean that that mutation was directly involved with this motility phenotype. We were lucky enough to find that one such mutation in a hypothetical channel protein, but even having that protein doesn't mean anything really, but to make sure it made some kind of sense with what we know about cholera biology. Cholera swims due to its single flagella, and this is only able to rotate because of an intact sodium gradient. If there are more sodium ions in the periplasm of the bacteria than the inside of the bacteria, the motor proteins of the flagella are allowed to shoot these sodium ions across, and this releases energy which allows the flagellar motor to turn. We are hypothesizing that this channel protein, which is embedded in the inner membrane, is very sensitive to antibody binding. Upon antibody binding, this channel can open, which would allow sodium ions across freely and cause the flagellar motor to stop swimming in the time frame that we're looking at. If this is true, this would be a widely applicable model of antibody-mediated motility arrest, and it can be applied to other bacteria like Salmonella species, which are another public health threat in the US as well. This information could directly impact what we know about vaccines at this moment and treatments, and could help break this cycle of cholera transmission and cholera disease, which is causing so many deaths in the world. Thank you.